Have you ever just stopped and wondered how something as complex as your brain can be built from just a handful of cells in an embryo? It seems impossible, right? Well, it all comes down to this incredibly elegant signaling system, a kind of cellular conversation that we're going to decode today.
I mean, this is it. This is the big question in developmental biology. For more than 100 years, scientists have been puzzling over this. How does all this amazing, intricate order just emerge from what looks like a totally uniform group of cells? The answer, it turns out, is in a series of tiny molecular conversations where cells are literally telling each other what to become.
So here's how we're going to piece this mystery together. First, we'll look at the default neural plan, kind of like the brain's basic blueprint. Then, we'll zoom in to see how a few specific cells get chosen from a crowd, like finalists in a competition. That's going to lead us straight to the main event, the notch conversation. We'll see what happens when that conversation goes wrong, and finally, we'll see how this exact same system is a fundamental building block for pretty much every animal brain on the planet, including ours.
Okay, so our story has to start at the very beginning. The first big decision an embryo makes on its way to building a brain is deciding which cells even get to be part of the nervous system in the first place.
The technical term for this is neural induction. Think of it as the first major fork in the road. A specific patch of the embryo's outer layer, the ectoderm, basically gets a new set of instructions. All right, you guys, you're now going to form the brain and spinal cord.
Now, what's really wild is how scientists figured this out. In these classic experiments, they took some of these ectodermal cells and just grew them in a petri dish, all by themselves, away from any other cells. And they didn't become skin. They spontaneously became neurons. It was this huge revelation. It turns out a cell's default factory setting is to become neural.
So, that raises a pretty obvious question, right? If being a neuron is the default, why doesn't the whole embryo just turn into one giant brain? Well, the answer is there's an active stop signal. A protein called bone morphogenic protein, or BMP for short, is constantly washing over the ectoderm, actively blocking that neural fate and telling those cells to become skin instead.
Okay, so to build a brain, the embryo has to find a way to shut down that stop signal, but only in one specific area. And this is where a little region called the organizer comes into play. It starts pumping out these proteins, like noggin and cordon, which are basically BMP blockers. They just soak up all the BMP in that one region, effectively taking the foot off the brake and letting those cells follow their default path to become the nervous system.
All right, so the brakes are off. We now have this whole field of cells that all have the potential to become neurons. But a brain isn't just a big clump of neurons. It's this incredibly precise pattern. So the next big challenge is, how do you select which cells become the neurons and which ones become something else?
Let's take a look at this slide, which shows us how this works in the fruit fly. On the left, we start with what's called a proneural cluster. You can think of this group of cells as a bunch of finalists for a job opening. They're all equally qualified, but through this really cool competitive process, only one cell gets selected to become the neuron, what we call the sensory organ precursor.
And this brings us to our next major problem. If all of these finalist cells have the green light to go neural, what's stopping them from all doing it at the same time? How does the embryo pick just one winner from the group? Clearly, there has to be another, much more refined layer of control going on.
And that refined layer of control is this absolutely beautiful cellular mechanism called the notch signaling pathway. It's essentially a direct cell-to-cell conversation that sets up a winner-takes-all system, and it makes sure only one cell comes out on top.
The process that Notch uses is called lateral inhibition, and the name is actually perfect. Imagine you're in a race. As one runner starts to pull ahead, they can literally reach back and tap their competitors on the shoulder and tell them to slow down. That's what's happening here. The cell that starts to become a neuron actively tells its direct neighbors, nope, you guys stop.
So how does this actually work at the molecular level? Well, it requires a direct physical conversation between two cells, right next to each other, just like you see in this diagram. One cell is the signaling cell. That's our future neuron. The other is the receiving cell, the one that's going to be told to stop. And what's really important here is that this isn't a long-distance message, it's a contact sport. The cells have to be touching.
Let's just walk through the steps here. First, the cell that's getting a slight lead starts putting a protein on its surface called delta. Think of delta like a hand. It reaches out and physically grabs onto the notch receptor on its neighbor. This handshake triggers an enzyme to literally chop off the inside part of the notch receptor. That little fragment, called notch ICD, then travels into the neighbor's nucleus. Once it's in there, it turns on a set of genes called HES genes. And here's the punchline. HES proteins are repressors. They are an off switch. They go and shut down the exact same genes that would have let that neighboring cell become a neuron too.
Okay, so we've seen the molecular nuts and bolts, but what does this actually look like in a real living thing? What happens when this super elegant conversation goes wrong?
So it all just boils down to a perfect balance. Like this slide shows, if you get a mutation in the pro-neural GO genes, you end up with too few neurons. But if you break the notch-stop pathway, you get the exact opposite problem, this massive overproduction of neurons that gives you that crazy bristly fly.
Now you might be thinking, okay, that's a neat little trick that flies use to space out their hairs, but the reality is so much more profound than that. This winner-takes-all system is one of the fundamental pillars of how complex animals are built.
This whole delicate dance, this push and pull between the go neural factors and the stop neural notch signal, it's an ancient strategy that evolution has used over and over again. This same molecular logic is used to build nervous systems in everything from insects to fish to you and me.
In vertebrates, like us, the system is adapted to do something a little different, but just as important. In our developing spinal cord, which you can see here, notch isn't just about picking one winner from a small group. It's used for timing. It tells a bunch of these neural stem cells, the ones in red, to just wait their turn. This makes sure we keep a big pool of these progenitor cells around, so we have a steady supply of raw materials to build our super complex brain over a long time.
And this leads to a result that seems totally backward at first. As this quote on the slide points out, if you get rid of the notch-stop signal in a mouse, you don't get more neurons, you actually get fewer. I mean, how is that even possible? Well, it's because without notch telling them to wait, all the stem cells become neurons all at once, in one huge premature burst. The whole pool of stem cells gets used up right at the beginning, and there's nothing left to build the rest of the brain.
So let's just tie this all together. We started with this cool idea that becoming a neuron is the default plan for a cell, but it's held in check by signals like BMP. Once that brake is released, a much more precise mechanism kicks in, that notch and delta conversation. This whole system of lateral inhibition lets one cell win the job of becoming a neuron while telling its neighbors to wait, making sure we get the perfect number and pattern of cells to build a working brain.